Hello, I am Álvaro del Real from the University of Cantabria in Spain, and I have just deposited my PhD thesis under the supervision of Professor José Antonio Riancho. My PhD thesis is focused on different epigenetic mechanisms regulating human mesenchymal stem cells isolated from osteoporotic patients. Osteoporosis is a skeletal disease characterized by an imbalance in the bone remodeling process, with a predominance in the bone resorption rather than in the bone formation activity. This leads to a decrease in the bone mineral density and consequently to increase the susceptibility to fractures. In this process, human mesenchymal stem cells are the progenitors of osteoblasts, which are the bone-forming cells. And nowadays, we don't know all the pathogenic mechanisms involved in this disease. So, a specific genetic change or a specific mutation is associated with multiple possible manifestations of a disease depending on the genetic background, the lifestyle, diet, environmental conditions, and all of them with epigenetic factors. Epigenetic mechanisms are referred to a number of processes that permit reversible modulation of gene expression without altering the DNA sequence to rapidly adjust cellular processes to constantly changing environmental conditions. Among epigenetic mechanisms, in my thesis, I have worked on DNA methylation and long non-coding RNAs. Both are very state and tissue specific. So the main objective of this work is to study the change of these epigenetic marks in human mesenchymal stem cells isolated from osteoporotic patients, and how these changes can affect the osteogenic differentiation and the bone forming capacity of these cells. So next I will proceed to explain the results obtained in the study. So first, we have characterized the functional capacity of human mesenchymal stem cells, which may be compromised in the elderly. Human mesenchymal stem cells from osteoporotic patients can have an altered differentiation capacity when cultured in osteogenic differentiation medium. However, they appear to maintain the proliferation rate. Then we have analyzed the DNA methylation and the transcriptome of the human mesenchymal stem cells, and we have shown that differentially methylated sites, when compared with our controls, were enriched in enhancer regions. Moreover, these genes with differentially methylated enhancers were overrepresented among those showing differentially expression, and gene ontology enrichment analysis revealed that these genes are related to bone pathways. Apart from that, concerning the non-protein coding gene expression, almost the 72% of all non-coding RNAs differentially expressed were of the antisense type of long non-coding RNAs. And the half of the protein coding genes in the cis position of the differentially expressed antisense long non-coding RNAs were also differentially expressed and overrepresented in bone-related pathways. So overall, our results suggest that epigenetic mechanisms, specifically DNA methylation in enhancer regions and long non-coding RNAs, play a significant role in the gene expression pattern of these human mesenchymal stem cells and may represent new targets to enhance bone anabolism in patients with osteoporosis. Thank you.